I am a microbiologist. I came here and I was given the opportunity to develop a class from the ground up, so create a new class. And I thought to myself, well, I love astrobiology, so I'm going to take this opportunity to develop an astrobiology class, and that's what brings us here today. So I teach ES 9002, which is Introduction to Astrobiology, and we consider the potential for the existence of life in places other than planet Earth. And so what I hope to do today is give you an introduction to this field. Okay, so in today's seminar, I'll give a brief outline of the following topics. So first, we're going to start off thinking about what is life. So our goal is to search for life in the universe, in the galaxy. Will we know it when we see it? So we're going to talk about 6 key characteristics that we can recognize in life on Earth, and that will help us develop a framework for what to look for when we look for life outside of Earth. Then I'll talk about the concept of habitable zones around stars. This is based on the building blocks of life that we use on Earth with the solvent liquid water. I'll talk about a few discoveries, our work to look for extrasolar planets in planetary systems and stars beyond our own, and the nature of some of these exciting new exoplanetary discoveries. And then we'll wrap up with a consideration briefly of some upcoming missions. Okay, so what's astrobiology? It is a combination of fields dedicated to look for extraterrestrial life in the universe. So in my class, I tell my class that no field of science is off limits. We cover nuclear chemistry, particle physics, astronomy, cosmology, biology, even like radiation toxicology. So really, it's everything that encounters life. We apply it to thinking about life in other systems. And so research in astrobiology primarily focuses on 3 major topics. One, studying the conditions that can give rise to emergence of life and ongoing existence of life. Second would be looking for those types of conditions on other planets in our own solar system and also around other stars. And thirdly is looking for the actual occurrence of life elsewhere. Now, you're looking at a picture of Mars. This is taken from the Curiosity rover. It's Mount Sharp in Gale Crater. And so, I mean, it's a really astounding image. It looks a lot like Earth, doesn't it? So we're exploring Mars right now. We're exploring a number of planets in our own solar system. And so when we do these explorations, how do we begin to find evidence for past life or perhaps current life? And one thing we can do in astrobiology is look at analog environments on Earth. So this is a picture of McMurdo in Antarctica, the McMurdo dry valleys. And so this is considered a Mars analog environment. And so scientists go here and they try to understand the type of life that survives there. And guess what? There's no trees, there's no animals, not even penguins in the dry valley. This is the life is primarily microbial here. And in fact, it's life that lives under rocks, even within rocks. So very microbial. And so that's a good model for what we might expect when we go to places off world that look similar to this. All right, so let's think about some of these characteristics that sort of define life on planet Earth. So there's a scientific consensus that a number of characteristics are shared, and the framework I'm going to share with you kind of summarizes these into a set of 6. And you might see other folks who say there's a set of 7 or a set of 4, but roughly they're talking about the same thing. So this framework is a set of 6 characteristics, and they are that life contains order and organization, that it is able to reproduce, that life can grow and develop, that it can respond to the environment, that it can adapt to its environment through evolution, and that it utilizes energy to do all of these things. So let's just look briefly at a few examples for these concepts. So order and organization. When we look at life on Earth, we see that there's structure down to the subcellular level even. So you're seeing right here, this is a up close view of a pine tree embryo. And so each one of these squares here is a plant cell that's at about 50 microns in dimension. And inside of the square, you see a little red dot, and that's actually the DNA or the nucleic acid, the genetic material of the cell that's been stained. So what you can see right now is that at the cellular level, there's organization. Even at the subcellular level, there's organization with the DNA in that enclosure. And so this plant embryo then will grow up to be a pine tree. And so the pine tree has even more levels of organization. It has woody structures and leafy structures. So that means that these tissues, then the tissues that are made up of the cells differentiate to form these other structures. And so that brings us to consider some of these other definitions of life. Okay, so the first one was order and organization. Life has order from the subcellular level all the way up to the manifestation of a whole body plan. Now, life forms reproduce. And if life forms didn't reproduce, then they die out, and an individual, an individual life form, when it dies, becomes the end. There's no more life. But because life forms can reproduce, they can survive the death of individuals and continue having a population. And so you've been looking at bacterial reproduction down here at the bottom of the slide. And so here bacteria, the very simplest life forms, they simply divide by growing and then splitting off, called binary fission. Now, organisms that are living also grow and they develop. So here we're looking at two pictures, one of a bean sprout growing. So you can see this is a pretty typical, you see the roots extending, then the little sprout pushes its way up, the leaves extend. So this is a clear indication of growth. But you also have programs of development that aren't necessarily growth. So now on the right, you're looking at a salamander egg. You can see that it starts off as one cell, and then you have individual cells that begin splitting and splitting and splitting. It's not growing in size, but it's developing. And it's developing into distinct tissues that'll grow out to be a salamander leg and a salamander arm, a salamander head. And so that's what you're watching take place here. So growth and development is a hallmark of living things on Earth, that they grow and then they also develop and can differentiate into some of those ordered structures that we saw in the pine tree, or for the salamander, it's different tissue types, its brain, its heart, its lungs, its appendages. All right. So, oops. Now, another key characteristics, or rather two key characteristics that living organisms share, the ability to respond to their environment. And they do this on short time scales via behavior. And then they also do it on evolutionary time scales. So that's called evolutionary adaptation. And so a good example of response to the environment is the migration of birds. So for example, in temperate regions, you'll have birds when it gets to be winter, they'll say, okay, I'm out of here. This is way too cold. I'm going to fly south, or I'm going to fly north where the temperature is nicer, and I'll have a nice time where it's not going to be so darn cold. So that's a response to the environment. They pick up a cue that it's about to get cold and they leave town. I don't know if any of you have seen in the news recently, but there's been an article about these tuskless elephants in Zambia, basically a nature reserve that had been subject to a lot of poaching over the years. And in just a handful of elephant generations, now there's suddenly a lot more elephants that just naturally don't have any tusks. And so that's because they haven't been culled by the poachers. So now we have this population emerging that has no tusks. It's a tuskless population. And so this is an example of evolution in action. And one of my favorite movies, Jurassic Park, the first one, Jeff Goldblum, says very convincingly, life will find a way. So this is an example of life finding a way. If having tusks means that you get slaughtered, then elephant populations in an evolutionary context respond by increasing the numbers of that population that don't have tusks. Another indication of widespread evolution on Earth is just the diversity of physiology in organismal types. So I'm a microbiologist, I study microbes. And one class of microbes that we look at are thermophiles. So these are microbes that can tolerate really high temperatures. We might be more familiar with microbes that are pathogens, that live at our body temperature, or that might make your food go bad, that might live at 20 degrees C or so. But there's actually microbes that can live in boiling water. So here's an example in Iceland. This is a hot spring that's at 100 C. Yes, there's microbes there. You go to the deep sea, you even have water that's superheated because it can be higher than the boiling point because it's so deep, it's under pressure. So microbes that can live at over 121 degrees C. And this is a beautiful picture. This is Grand Prismatic Spring in Yellowstone National Park that's in the US. And so this is a super heated geyser. It's at 74 degrees C. And then you can see these algal mats all around it in bright orange and some other mats here in yellow. And so these are microbes that are algae. They harness light energy to grow, and they're living in this superheated environment. And then in our own backyards, we have now the Sembawang Hot Spring Park. Sembawang Hot Spring, of course, has been around for a while, but it was recently renovated and opened in 2019 as a hot spring park. And so we have our own backyard thermophiles growing in Sembawang. So these thermophiles are an example of organisms that we like to refer to as extremophile, where the Latin root of this term, phile, means to love. And so there's lots of different types of extremophiles. We have microbes and even some macrobes on Earth that can thrive under really high pressure conditions. These are barophiles that can thrive under highly acidic or even highly alkaline conditions, acidophiles or alkalophiles. We have organisms that can survive really high concentrations of solutes like brines, and these are osmophiles. You can read the list here, but many different types of organisms that can survive really diverse types of environments. And appreciating this diversity of environmental conditions under which life can survive helps frame our search for life in the solar system, because it's not just a narrow set of environmental conditions, it's actually a pretty broad set that we can now look for elsewhere in our solar system to find analogs. Okay, so that was a long bit on evolutionary adaptation as the fifth example of a hallmark of life on Earth. So the sixth one is energy utilization. So all of those things we've just been talking about require energy. And organisms, they need to consume energy to carry out the necessary steps for these key requirements of life. And organisms obtain this energy through a process known as metabolism. And metabolism is something that happens in the cells. So on Earth, we consider two major types of metabolic types. We have the organisms that get their energy from light, from sunlight. These are called phototrophs. So I'd like to go back to the Latin just to make sense of these science jargon terms. Troph means to eat, or one who is nourished rather. And photo means one that's nourished from the light, right? So phototrophs are organisms that get energy directly from light. In other words, plants. You're like, why didn't you just say plants, Professor Thompson? Well, okay, so plants, but we also have our algae, our cyanobacteria, so organisms that grow based on light energy. And then on the other side of the coin, we have organisms that get their energy from chemicals. We're organisms, we get our energy from chemicals. These are chemotrophs. They get nourished from chemicals. And so these chemicals can be organic chemicals, like when we eat food, it's organic. That doesn't mean grown chemical free. We're not talking about like food labels. I'm talking about organic chemistry. So organic means it's carbon rich. So we can have carbon rich food, or we can have inorganic food, which would be basically no carbon. So things like sulfur compounds or reduced metal compounds can also nourish some microbes. So microbes can get their energy from these types of chemical compounds. And so the example I give in my class is the chemotroph Michael Phelps, won 23 gold medals. And I was just so impressed by this infographic on his diet. That's a lot of healthy eating for winning Olympic gold medals. But so Michael Phelps is an example of a chemotroph. You and I are chemotrophs. Okay, so we've seen these 6 key characteristics that are shared by living organisms. And it's thought that these would be shared not only by living organisms on Earth, but also elsewhere. So we saw order and organization, reproduction, growth and development, response to the environment, adaptation, and energy utilization. And so these are necessary for life individually, and then in a combination, they would be sufficient for life. So how do you build a life form? Well, you need building blocks. And so these building blocks create the biomass of a life form, making a compartment for the processes of life to occur, like metabolism. You also need a liquid, and this liquid acts as a solvent so that things can move around and circulate, reactions can happen, waste can be removed. So on Earth, carbon is the major building block and water is the major solvent. And I'm looking at the time here. So I need to be wrapping up by what, by 8:15? Maribel?
Yes, roughly.
Yeah.
But it's okay if we go a bit longer.
Okay, well, I'm aiming for 8:15. Okay, so so when we look at the building blocks that we see on Earth life, we see that carbon is that building block. And if we look at the distribution of elements in the universe, and this is so cool, you can see that carbon is one of the most abundant elements in the universe. This is an element that's made by stars. So the first two most abundant elements are helium or hydrogen and helium, followed by oxygen, and then carbon, and then nitrogen. So these tables over here show you the percentage of these building blocks in a human, percent by mass, and then percent by mole, atomic percent down here. And you can see these most abundant elements in the universe are the same building blocks that make up life. Is that a coincidence? I don't know. Okay, so carbon is a particularly great building block because it can form these really nice, long, stable chains. And it can have a lot of structural diversity in the types of molecules it makes, which means that it's a really good building block for creating the biomass, the structures of life. You can think about some other building blocks like silicon and nitrogen. They have some deficits when you're thinking about making biomass. For one, silicon, for example, it doesn't form as strong bonds. It doesn't have the same sort of diversity of structures that it can make. So for example, it won't form double bonds. Nitrogen, it also forms very weak nitrogen-nitrogen bonds. You can't really make long chains. Also, it has the side effect of being highly combustible due to kind of the fact that nitrogen bonds are very weak or very, very strong. But people really like to think about these other types of building blocks for life because it really opens our mind to thinking about more inclusive definitions for life. Life with a Y, we sometimes say. And so life with a Y, this kind of life using other building blocks, is currently unknown to science, but it's really interesting to keep in mind as we think about exploring other worlds. That said, carbon still seems like it's the winner when it comes to building biomass. Now, what about solvents besides water? So all life on Earth is water-based, but could another fluid possibly fill the role of water? So what you're looking at here is a table of potential solvents that have been considered for life with a Y. So we can see compared to water, we are familiar freezing temperature 0 degrees to 100 degrees. It's got 100 degrees C liquid range. So that's a wide window, and it's also a pretty wide window that includes temperatures that allow pretty fast kinetics. If you look at a lot of these other types of solvents, either the windows can be a bit narrower, the exception being methanol, of course. So the window where the fluid is liquid is a bit narrower, but they tend to be a lot colder, which means any sort of kinetic process in them would be a bit slower. All right, so again, we don't know any life forms like the Y that use these alternate solvents, but water is the solvent of choice on Earth. And water is pretty great for a number of reasons. First, we saw that broad liquid temperature range that includes the warm temperatures that allow fast chemical kinetics. But my favorite one is that ice floats. And this is not the case for all ices. So water ice has the unique property that it's less dense than the liquid, and so it floats, which means that you'll have, if you have an ocean, you have a nice insulating layer of ice. If you look at ammonia or methanol, for example, those ices will sink. And if you have an ocean that's comprised of one of these ices, it will basically freeze in the colder outer layers and then sink, building up a frozen solid. So it would be difficult to maintain a liquid phase. You could very easily have like frozen solid oceans. And then lastly, water participates in a lot of our chemistry. So there's charge separation in the water molecule. It's very polar. And so it plays a role in our metabolism. Okay, so liquid water, we've established that it has some key advantages. So it's no surprise that Earth life has liquid water as its solvent. So as we start to think about our search for life in other environments, focusing on where liquid water might be makes a lot of sense. And so that search is kind of organized into the framework of habitable zones. And a habitable zone is the range of distances around a star where worlds hosting liquid water on the surface could exist. But simply being an object within a star's habitable zone is not sufficient to have liquid water. For example, our moon doesn't have any liquid water on it, although it's in our sun's habitable zone. So you need other factors as well, like an atmosphere, the ability to retain an atmosphere to keep your surface liquid water in place. And also we can note that stars, as they age, will grow more and more luminous, which means that their habitable zones will grow outward with time. So if we look at stars in the night sky, and this image might look a little familiar to you because it's my background here, you see a really wide diversity of stars. And I don't need to tell the Astronomy Society about this, but I'm a microbiologist, so I'll say, wow, look at all the lights. It's great, right? You've got blue, you have reds, you have yellows and oranges. And so a lot of these stars would be on the main sequence, but some of these stars would be stars that are at the end stages of their life. So in particular, some of the really, really bright red ones, the large red ones, those would be giants. But a lot of these stars are on this, what's known as the main sequence. And so this is an illustration of this main sequence. I think some of you must be familiar with this, but let me just go through it. So you have luminosity here on the y-axis, and then you have the surface temperature of a star on the x-axis. And so you see this linear relationship between luminosity, where the brightest stars are the hottest stars, and the least bright stars are the coolest stars. And this also corresponds to the star color. So what we see is when looking at the distribution of stars, that 75% of the stars that have been observed and are known to be on this main sequence are these M-class stars, these very dim stars that shine most brightly in the red and in some cases the infrared wavelengths. So these are the M-class stars here. Then we have the K-class stars here. About 15% of the objects are K-class stars, and then about 7% of them are G-class stars, including our own sun. And then the balance, about 3% or so, are these other classes that shine towards the white and the blue. And the ages of these stars, so the maximum lifetime, also correlates with these axes, where the M-class stars actually live the longest. They live hundreds of millions of years, whereas stars like our sun will only live 10 billion years. And then some of these supergiants will only live a few hundred thousands of years. And so this has some important implications for thinking about the emergence of life. So let's look at how these different star types influence the habitable zones around them. So we're only going to think about the stars that last for 10 billion years or longer. So we'll start off with the G-type stars here. So this is our sun, this is the example. And so here we can see that our habitable zone is predicted to include Earth, fortunately. Also, depending on the model, including Mars at the outer limit. And as our sun ages and grows brighter, eventually the habitable zone, or Earth's orbit will fall out of the habitable zone because the habitable zone will keep moving out. But that won't happen for 500 million years or so. Okay, so we can see the G-type star. It has a pretty distant habitable zone because the sun is more luminous, it's giving off more heat. And it also, this distance affords some protection to the radiation that the star gives off. Now, if we go down in size, we can consider the other extreme, the M-type stars. And so these are the smallest, the least luminous objects, but they also have habitable zones, zones around them that can support liquid water on the surface of planets. But these habitable zones are far narrower, right? Because there's just not as much heat being given off by the stellar object. And they're much closer to the star because they're getting, I mean, basically they're just going where the temperature is, right? That's the habitable zone is defined by the temperature, and cooler stars, you need to be much closer to them to have that same level of heating. So these stars are really interesting to think about in terms of the search for life because they last for so long. Oh, I have a typo on this slide, I'm very sorry. So these stars do not last for hundreds of millions of years. They last for hundreds of billions of years. So that should be a B, over 100 billion years, these M-type stars. So that's a lot of time for life to emerge and to evolve. But the downside of these stars is that the habitable zones are quite close to their stars, which means that the planets in them would be subject to high radiation flux. And of course, radiation is not great for life. So radiation damages biological tissue, causing it to degrade, and it's thought that organisms really wouldn't survive very well in environments with really high radiation. Now, these K-type stars are kind of thought to be in a bit of a Goldilocks zone when you're thinking about habitable zones. These K-type stars, which last a bit longer than our sun, so on the order of 20 to 50 billion years, their habitable zones are a bit wider, they're a bit further away from their stars, and so they're less in the path of stellar flares and radiation. Okay, so there's been a lot of really interesting research and discoveries coming out over the past decades. The first exoplanet was discovered in '95, and as of today, there's over 4,500 exoplanets that have been discovered. And some of these worlds have even been directly imaged, and this requires some pretty fancy optical methods where you have to block out the light from the center star. We're going to see an example of that in a second. But most of these planetary systems have been discovered through indirect observations that either use changes in brightness of the star or the gravitational interactions where the star will wobble as a planet and it orbit around a common center of mass. So we'll look at two examples of how these are done. But first, let's take a look at the data on the discoveries. So this plot gives you the discovery year here on the x-axis. And then on the y, you have the number of detections. It goes up to about 1,500. So you can see that most of these detections are green. So green corresponds to detections made by the transit method. And so that's the fluctuation in brightness method. And coming in second is the radial velocity method. And this is the method where you're looking for the wobble of the star as it orbits with the planet around a common center of mass. All right, so let's look at some examples. So these are planets that were discovered by direct imaging. And here's what I showed you where you can block out the light from the center star. And it just so happens that these beautiful objects here are big Jupiter-like planets, and that the kind of configuration of how they orbit works just right. And so this is a movie that's made from stills taken over about 7 years. And you can see these planets so well because they're just gigantic planets. Okay, so only a handful of discoveries have been made this way, though. It's not easy to get this sort of imaging. Now, by far the vast majority, so all those observations in green, have been made by the transit method. And so here we're going to look at an animation of the transit method where you can follow this plot here. So basically in the transit method, you're looking for dips in the brightness of the star as a planet passes in front of it and blocks out some of its light. So the biggest planet will have the biggest dips, and smaller planets have smaller dips, but they can be superimposed on each other. And so then from these sort of light path data, you can back out both how many planets are passing in front of the star, as well as determine their orbital period or the length of their year. Okay, and then the second method is the Doppler method or the radial velocity method. And here, like I said before, you're looking at those gravitational wobbles. So as the star and a planet go around a common center of mass, you'll have periods of time where the star is moving away from you and periods of the time where the star is moving towards you. And of course, the angle has to be just right. Only a minority of stars are actually in the right angle for you to get that Doppler shift information. But you can use it then to understand the orbital period by the frequency of that radial velocity change. Okay, so this is a graph. I got this just a few days ago from the Caltech Exoplanet Archive, and it shows the discoveries to date based on the transit method. Well, I'm sorry. It shows the discoveries to date that will provide a radius for the planet. So these are all the planets that have been discovered that have a radius estimate. So they have an estimate for how big around it is. And surprise, surprise, most of the planets with radius measurements were discovered by the transit method, right? Because the transit method will tell you how much light is blocked, which you would relate then to the planet size. And another major bias in this data is that most of the measurements have periods between 1 and 100 days. And that's kind of due to the nature of the observing, where a lot of these observation windows aren't really that long. You're not going to just watch one system for years and years. So, a lot of the data is biased towards these shorter, more frequent transits. Now, if we look at the planets for which mass data is available, you can see that there's actually a lot more diversity in terms of the periods that have been revealed. And so some of these radial velocity measurements, for example, have detected planets that have orbits on the kind of same orbital period as Earth's year, right? So the key is that the more methods that we can use to detect these exoplanets and to discover them, the more diverse exoplanets we'll be able to discover. Now, you might have noticed that planets that are detected by both the radial velocity measurement as well as the transit both have the same orientation, which is that sideways view. And so that means that if you find a planet by one method, you can then confirm its existence by another method and give yourself some complementary data. So if you have both mass and size of a planet, you'll have the density. So the density of a planet lets you predict whether it's rocky, like an Earth-like world, or whether it's a very light density, like a Jupiter, a gassy giant. So this is a plot of the density of exoplanets. And this plot is a little dated. It's from 2015, but I couldn't actually find a newer one. So this is what we've got. It plots radius on the y-axis with mass on the x-axis. And then you see these lines. These are lines of constant density here, where the density would track foam. And you can see my pointer, right? So it tracks foam here. The density tracks water in this middle line, and then the density tracks lead in this last line here. So our solar system planets are these green dots. And you can see that they fall kind of close to the water line, although down here you see the terrestrial planets, Earth and Venus, that fall between the density of water and lead. But looking at Uranus and Neptune, even Saturn and Jupiter, they kind of fall on either side of this water line. If we look at the densities of the exoplanets that have been discovered, they're a lot more all over the place, a lot wider distributions. And so some new classes of planets have actually been discovered. One of those classes is hot Jupiters. And so these are gas giants that during the formation of their solar system have migrated inwards and so are currently sitting next to their suns, next to their stars. And so because the Jovian planet is sitting next to the star, its atmosphere expands even further, becoming even less dense than our own Jovian planets. And so the name given to these are hot Jupiters. Another type of planet that has been discovered through exoplanet surveys are these super Earths. So these are these rocky terrestrial type planets that are a bit larger than Earth. And then we have mini Neptunes, which would be, as the name infers, Neptune-like planets that are smaller than Neptunes, kind of intermediate between Earth and Neptune. And so these would have very thick atmospheres and possibly, likely to be enriched in hydrogen compounds like water, ammonia, and methane. Okay, these are just some examples of super Earths down here. And we have the mini Neptunes here. We have some hot Jupiters over here, very dense, light, low density planets. Okay, so exoplanets, a bit more diverse than the planets we know and love in our solar system. But taken together, we do find a good number of Earth-sized planets. And in fact, it's been estimated that about 70%, 50 to 70% of the planets of the stars that have been studied have planets around them. And a good chunk of them are Earth size or super Earth size, which bodes well for our exploration for Earth-like worlds. There's a group actually that focuses on ranking these exoplanetary discoveries by their Earth similarity. And they've developed an index called the Earth Similarity Index, or ESI, where 1 would be the same as Earth, and 0.9 would be very high, indicating 90% similarity. And so here they've ranked some of these exoplanets, and they're organized based on their proximity to Earth. So 2 interesting ones I'll just point out here. T-Garden star B, 0.95% Earth similarity, and then Proxima Centauri B. So Proxima is interesting because it's our backyard exoplanet. It's only 4.2 light years, 4.1 light years away from us. This is the rank list. You can see lots of cool data. So if you ever want to check this out, just go onto this website here, the Habitable Exoplanets Catalog. And so it ranks these planets. It gives you the type of star. You can see most of these stars are M-type stars, a few K-type stars, 1 G-type star. You can see their mass or their radius based on the type of measurement that was used to discover them. And then the predicted equilibrium temperature of the planet, and then the ESI score. So just a few quick notes. T-Garden star B, the highest similarity star. And we don't know a lot about these stars, but this orbits an M-type star. It's 12 light years from Earth. And in the system, there's actually 2 potentially habitable exoplanets. And the discovery was made by the radial velocity measured. Now, one consideration is, since these planets orbit an M-type star, the habitable zone is very, very close in, which means that the planets are likely what's called tidally locked, which means they're kind of like our moon. One side will always face the star, and one side will always face outwards from the star. So that could potentially limit the ability to host liquid water. So yeah, there's a cool diagram of this system. And then our friendly neighborhood exoplanet, Proxima Centauri B. Again, it orbits another M-type red dwarf star, very, very close to home. There's actually 2 planets that have been discovered in its orbit. Proxima Centauri B orbits very close, 0.05 AU. So 1 AU is the distance between the sun and the Earth. So 0.5 AU means it's 1/20th of the distance between our sun and the Earth. So it's very, very close into its star. Again, probably it's tidally locked. Proxima Centauri B, the star Proxima Centauri, has a lot of magnetic and flare activity, so it's probably completely fried, but who knows? We don't have the best data yet, so we're still very carefully studying it. So the second planet in this system, Proxima Centauri B, is thought to be kind of a super Earth size, mini Neptune size. So it's 7 times as massive as Earth. And see down here, it orbits once every 5.28 years. And there's actually some evidence that there might actually be a ring system around this planet. So it would be really amazing to potentially visit the system someday and learn about another solar system. So speaking of visiting Proxima Centauri, let's talk about future missions, and this will be my last section. So upcoming in December is the launch of the James Webb Telescope, and this is really exciting for astronomers and astronomy-friendly people all around the world. So the James Webb Telescope will have a lot of really cool instrumentation on it that will let it take really, really sensitive measurements in the infrared, near infrared, close near red-ish colors range. And this is great because a lot of the stars that we are interested in looking at the exoplanets are, radiate very, very highly in the red and the infrared wavelengths. And so the James Webb Telescope has a lot of modifications to make it really well suited to observing in the infrared wavelengths. The first is this big sun shield. And so this is not just a sun shield, it's also an Earth shield and a moon shield. And so what this does is it blocks out any infrared radiation signatures coming from the sun, the Earth, and the moon. And it can keep the sun, the Earth, and the moon on its shield side by its targeted location. So it's going to be positioned out here at the L2 point. It's a Lagrange point. I'm not going to try to explain the physics of that to you, but it's a point that orbits around the sun, and it can maintain a constant position relative to the sun and the Earth and the moon. So it can always keep the Earth and the moon and the sun on the one side, block out their light. So this telescope requires like really, really cold conditions to operate. These are conditions even colder than the surface of Mars. We're talking like Pluto cold to be able to take these sorts of measurements. And so that's, the launch has been delayed many, many times, and most recently, of course, due to COVID, but the current launch date is sometime in December. So it'll be a fun Christmas present for those of us who like space-based telescope observing. And one of the nice things we'll be able to do with this telescope isn't just take images of worlds or to detect more worlds, but also to begin to study their atmospheres. And so we can use the light signatures that get transmitted through a planet's atmosphere to do spectroscopy. So some of the instrumentation on this telescope has the capability to do spectroscopy. And in the spectroscopy method, you're actually going to look for signatures from different elements. And if you can know what types of elements are in a planet's atmosphere, you can begin to make some models about what types of geochemical processes might be happening there, but also potentially biological processes. And this is just an example of a spectrograph that was obtained as light from a star was transmitted through a sodium-rich atmosphere of an exoplanet, a rather large exoplanet. And then it gave this absorption spectrum when you can normalize it to the signature from the star, and you can see, oh, this planet has an atmosphere that has a lot of sodium in it. So very cool technique and approach. The last the second rather future mission I wanted to mention to you guys is this kind of a bit more ambitious even than the James Webb Telescope. This is the Starshot to Proxima Centauri. And so this is a privately funded ambition. Got some millionaires on the board. I think Yuri Milner is the head one. And did I say millionaire? I'm getting my millions and my billions mixed up in this lecture. So these are billionaires, not millionaires. The initial private funding was 100 million to start research. The idea is to send very, very lightweight cameras on a 30-year journey to the Proxima Centauri system and take some great pictures of our close neighbor solar system or planetary system, and then beam those back to us. So this is just a quick animation of their ambitions here. So basically the idea is to create a laser array somewhere in the world, and then to have these little tiny cameras, 2 megapixel cameras, in the middle of a 4 meter by 4 meter sail. And then you're going to hit the sail with a blast of tons of laser energy and accelerate it to about maybe 10% the speed of light so that it can make that transit to Proxima within a reasonable time frame. Now, I mean, if an Earth government is going to let you do a laser array, that's a huge logistical hurdle that they'll need to sort out. I can imagine a lot of people being pretty concerned about a country having the ability to shoot down fire lasers into space, but still, you can send these cool little mini spaceships on this 4 light year journey. And the idea is that they'll sail past, I guess that would be Proxima and Alpha Centauri in the distance there. And, you know, we'll see what we see. We see beautiful, fertile worlds or kind of a radiated, blasted, barren landscape. It'll still be phenomenally interesting and, you know, groundbreaking. And now we know there's not just 1 planet there, there's thought to actually be 2 planets. So pretty neat, a very ambitious project, and it'd be really interesting if they carried it off. All right, so to wrap up then, concept summary. We saw that life, or life with a Y, could be described by these 6 key characteristics: order and organization, reproduction, growth and development, responding to the environment, adapting to the environment on evolutionary timescales, and then using energy. We learned about extremophiles, microbes that are lovers of the extreme, that challenge the boundaries of where life can exist on Earth, and it helps us understand the range of conditions where Earth-like life might flourish, both in our solar system as well as beyond. We learned about a star's habitable zone, which is the range of distances around a star that would allow it to potentially harbor liquid water. It's not enough to just be in the habitable zone, though. You'd also need an atmosphere in some way to keep the atmosphere and the liquid water in place. We can detect exoplanets by a number of methods. The most prolific has been the transit method. We also learned about the Doppler shift method. And we saw that transit method gives you radius, Doppler shift gives you mass. You put them together, you can get density. And density lets you look at the diversity of planetary types. Is it terrestrial? Is it rocky-like? Is it gaseous, low density, Jovian type planet? And current evidence is suggesting that most stars actually do have planets, and at least some of those planets are Earth-sized and in their star's habitable zone. And we saw this Earth Similarity Index, which is based purely on the planet size and the amount of light and heat that it receives from its star. But we can rank planets based on their Earth similarity. We saw 2 examples, Teegarden star and Proxima Centauri, as stars that host Earth-like planets. And of course, these planets can be prioritized for further research. And also, we saw that if the upcoming James Webb Telescope launch expands our opportunities to study the atmospheres of some of these exoplanets, where we can use spectroscopy of light, in particular red and infrared wavelengths of light passing through an atmosphere, looking for the fingerprints that chemicals will leave behind to understand kind of more of what's going on with the geochemistry and the potential biology of these worlds. So that wraps it up then. I hope you've learned something and it was to your interest. So thanks. I'd be happy to take any questions, but first I've got some MCQs for you guys. So maybe I could take one question, maybe while folks are getting signed up.
Yep.
Hi, Professor.
Hi.
I was actually, thanks for the great lecture. I wanted to ask, so basically, when you talk about the James Webb telescope, you're looking at near infrared wavelengths. So is that more towards the M-type stars because they're dimmer and darker and will emit more near infrared wavelengths?
I think that's definitely a benefit. The mission objectives of the James Webb Telescope are pretty vast, and I can't claim to be able to tell you about like all the different targets they want to study, but they determined that that kind of wavelength was the best for focusing on, and it's definitely going to be useful for looking at these M-class planets. Like the Spitzer mission that was retired not so long ago, it also did a lot of observing in the infrared, and it was able to discover so many new M-type stars and exoplanets around them. So yeah, so I think that's definitely a benefit of it. I don't know if that was the primary driver or not, but it certainly is a good thing for those of us interested in exoplanets and their atmospheres.
Thanks. Sorry, one more question. There was one slide where you were talking about the ranges of the liquid temperature, from solid to liquid. Does the range of the temperatures matter? Like, for example, for water, it was like 100 degrees Celsius to 100 degrees, to 0 degrees. And then for like nitrogen, I remember it was like something to something, but it was around like 163 degrees of range. So does that really make a difference in terms of
the range
amount of play?
tells you
Yeah.
how much an organism, how much environmental variability an organism can tolerate without freezing. Right? So the range is pretty important. How are we doing on signups? Are you guys ready to start the curtain?
Already we have 12 names here. So I believe that 19 people should play, or I think we wait for a bit more.
Can I take another question then?
Yeah. Actually, I have a question.
Sure.
So, so let's say when we send like rovers onto Mars, and I think it's always most likely the rover would bring like microbes onto the planet. And
I'm sorry, what did you say? Most likely it would be what?
It would bring like microbes onto the planet. Because I think it's very hard to eliminate all the microbes on the rover before you send it out.
Yes and no. I mean, I know the space agencies take great pains to minimize microbial contamination in the rooms where they assemble these rovers. And so you see people like in head to toe clean suits. So it can't ever be a perfectly sterile environment, you're right, but there's a lot of effort that goes into really reducing the microbial load before launch. And then, it's going to be in space without any sort of shielding from radiation from the sun. So that's UV light and ionizing radiation for, at least 8 months or so on the way. And so it's thought that can take care of, any sort of residual contamination. But at the end of the day, absolutely, there could be a couple of microbes getting to the surface of the planet.
So contamination to other planets is possible, but very low.
Yeah, and it's definitely an issue, a big issue that's on the radar of the space agency. And there's a planetary protection framework about how you manage that microbial risk. And so, the orbiters that went out to Jupiter and Saturn, for example, at the end of their lifetime, they were purposely destroyed by sending them into the atmospheres of Jupiter and Saturn so that they could avoid any risk that those would collide with the Jovian moons and potentially contaminate them. So, this is definitely part of the thinking in how they organize the space missions to manage that risk. I mean, they can't eliminate it altogether, but it is pretty carefully managed.
Okay, thank you. All right, I think we can start now. So those of you who have questions, you can either unmute yourself or you can type in the chat box.
So somebody, so Andy sent an article to the chat, and it's an interesting article. It's about the hypothesis that that planets that are kind of about the size of a mini Neptune, or maybe even a bit smaller, could have really strong greenhouse effects. And the way they'd have those really strong greenhouse effects is by having hydrogen, a lot of molecular hydrogen in their atmosphere. So Earth doesn't have enough kind of gravity to retain a lot of molecular hydrogen, but some of the more massive worlds can retain molecular hydrogen in their atmosphere. So yeah, it's very interesting kind of hypothesis for another class of world. So this would be a class of world that's somewhere between a super Earth and a mini Neptune. You know, basically they're saying radii as large as 2.6 Earths, yeah. So it's a great target of planets, and it'll be really interesting as the James Webb Telescope can you know, look at some of these targets that they've prioritized based on this hypothesis. And I like in the abstract, I'm sorry, I haven't read the entire article, but just from the abstract, they mentioned this idea of having tidally locked worlds. So these are worlds where one side would be facing their star. So in this case, presumably like an M dwarf star. And the habitable zone might actually occur on the side away from that side, star, or the side facing it. So thinking about sort of an asymmetric distribution of light and heat. Of course, if you have a really thick greenhouse effect from some great atmosphere around your planet, that atmosphere can help redistribute some of that heat. So even if it was tidally locked and you'd have a lot of intense radiation coming in from one side, you know, it'd certainly be a hotter side, but still the atmosphere could help spread out some of that heat around. So did you have something in particular in mind that you wanted to talk about this? Yeah, no, totally interesting article. And I think you see these types of planets on the density plot that I showed. You know, I mean, they're just such a range of different densities, right? So it probably fall kind of close to the waterline, I guess, because it's got rocky, but also a lot of water.
Can I ask one last one question? Just like a funny trick question type of thing. So like you said that you need 6 characteristics for life. Then you need order and organization, reproduction, growth and development, and all that. So what's the difference? And earlier in the question you talked about order is necessary but not sufficient. Sufficient. Is that right? So what's the difference between that and like a smart driving car? Well, a smart driving car has order and organization. He can reproduce itself in the assembly line. Then the development is in, yeah.
A smart driving car can't reproduce itself. A smart driving car could have you know, something else reproducing it. But okay, so let's say you have a 3D printer smart driving car.
Yeah, exactly. Yeah.
Can it adapt in an evolutionary context? So if it runs off of gasoline and all of a sudden there's no more gasoline, how would it accommodate that change? If it uses electricity, but suddenly it doesn't have access, or the plug type changes, right? Then how could it adapt? So can it adapt to changes in its environment? Does the reproduction mechanism that it have allow it to generate some diversity, where as conditions shift, it can also shift with the conditions? So
well, I mean, like if you say that, you compare that to like a human, if I'm suddenly thrown into the Arctic, then I wouldn't be able to like survive as well. So if there's like time for the smart car to the software to update, then wouldn't it be able to then solve that issue? And then wouldn't that make
you might have just created a cyborg?
Yeah, wouldn't that be alive? Would that count as being alive? And
so you do have algorithms that can evolutionarily adapt, right? So if you're combining an evolutionary adaptation algorithm to your smart driving car that also has a 3D printer backend that can make copies of itself, I think that's getting kind of close to life. What else have we missed? Can it grow and develop? So
you'll make a new model.
It's just kind of assembled but it doesn't really grow, right? But I guess that would be easy enough to program your 3D printer to continually have it grow, right? To take resources.
Make improvements. Yeah.
Yeah.
Then that's like the evolutionary adaptation. And then like
I mean,
it can respond to environment. Yeah. It can
nowhere,
respond to
nowhere
environment.
in this framework does it say it cannot be man-made, right?
Yeah. Then that's the only like fallacy it has. So yeah. I think that's just a funny thought that I had when I was looking at the 6 characteristics.
Yeah. That's good thinking.
Yeah, that's about it. Thanks.
I mean, there's some that would argue that planet Earth is a living organism because it also can evolve as a system, as an Earth system. There's some people that would argue that structures like emergent structures, like a beehive, that some of these like biological emergent patterns are also alive. So like I said, I mean, there's a lot of people who have thought about these frameworks for life. The 6 framework, the 6 characteristic framework that I use, I think it's a convenient logical framework, but there's other frameworks as well. They do touch on the same kind of ideas.
So like these are like the main ideas, but not necessarily the right framework under certain like for example the smart car that creates itself, that might not necessarily work for that. So you need a more specific characteristics that you can apply for that. Then you can disprove that smart car isn't a car. Is that what
But I mean, what's the advantage of trying to disprove that that's life? I mean, all we want to do is we want to be able to go to the ocean of Europa and send a camera down and some instrumentation. And if we see a kind of weird, funny looking shape, we're like, I wonder if that's life. Can we do some observations to see if it's life? So this framework helps us to think about that. Can look at it at the microscopic level. Does it have an order and an organization? If we observe it over time, does it grow? Does it respond to its environment? I mean, I don't know if we'd be able to observe evolutionary adaptation, but still some of these other things give us ideas for what to look at when we were looking at just new to science types of life forms. These give us some things to start looking for to characterize if it's life.
All right.
Like if you move it out of its environment, does it persist? Does it die? That's like a response to the environment, right?
Yeah, that makes sense. That makes sense.
So I mean, I think this framework is more for helping you recognize life rather than trying to say that androids don't have rights, right?
Yeah.
Well, that's a different class.
I understand.
Yeah.
Right. Thanks so much.
Right, so I would like to ask another question too. So I think we have all seen like all kinds of visualizations of extraterrestrial life, and they can look really bizarre sometimes. So is it reasonable to assume that life form will be very similar to what we have on Earth, on the exoplanets that are very similar to Earth, and would look much more different if they're on planets that are more different from Earth?
I mean, it's really impossible to say. There's a couple of things to keep in mind. One of those is this property called convergent evolution, where organisms that have very distinct genetic backgrounds that interact with their environment in the same way can tend to evolve the same sorts of traits. So for example, if you look at dolphins and sharks, one's a fish, one's a mammal, but they both have the same sort of body plan for their size, similar size. They have the same sort of body plan for moving through the water. So there's a number of examples like that in nature where organisms that interact with their environments in the same way develop similar body plans, even though they're distant. So in some sense, you might expect some of the similar features to arise when we look at aliens, if we do ever find them. But in other senses, it's going to be completely new and different because it's not going to be Earth, right? So, both dolphins and sharks live on Earth, right? And so there'd be a different set of conditions that would be kind of bespoke to whatever environment it was. Now, at the other side of the coin, though, most likely the first types of life we'll find are going to be microbes, right? I mean, microbes are what's in the Antarctic dry valley. They're very robust. They can occur over a wide range of conditions. When you get to the macrobes, the larger organisms, they have a bit narrower physiological tolerances. So, when you're talking about microbes, how might microbes look? Well, I guess it sort of also depends. We don't know what the last common ancestor of microbes on Earth was. We know that all life forms on Earth share DNA. They share some of these major molecules, and so they have a universal common ancestor between the organisms on Earth. Would that last, the universal common ancestor also be shared with other life forms elsewhere in the universe, perhaps? I mean, maybe there's some sort of a broad distribution of biology throughout the cosmos. We don't know that, right? So, I mean, I think that would also play a significant role in how things look like if they have DNA or if they don't have DNA, for example. And, microbes are microscopic, and, you can't really tell much difference between the two major domains of life on Earth, which are the bacteria and the archaea. They both just look like small little cells under a microscope. So at that level, I would expect that, extraterrestrial life might look similarly, right? Because microbes aren't really that charismatic to look at. So it might resemble terrestrial microbes, but still be something completely new and different.
So it's most likely that it will be very different from what we have on Earth.
I can't assign a probability. I mean, there's reasons to think it could be similar, right? But of course, there are also reasons to think it could be different. And my thinking at the end of the day as a microbiologist is that it will be microbial.
So we've been sending probes out with messages like the photos or something like the disc that plays songs, I think. So it's likely if the extraterrestrial life are microbes, they won't be able to understand it.
Yeah, I mean, I guess those kind of messages are really geared for intelligent life forms, right? That might be able to track down the signal and learn about us.
Thank you.
Sure.
Does anyone else has any other questions? If not, I actually have one more question who's, this is from our sign up form for this talk. And so this person asked, what are the obstacles in finding life outside our solar system?
I think access is a huge one. I mean, we can only do remote observation because of the distance involved. And so far, the remote observation, if you're going to look for microbial life, which is probably going to be the more abundant, more higher probability life form to detect. If you're going to look for that, you're really going to be looking for changes in the chemistry of the atmosphere that could indicate some sort of a catalytic process happening that could be biological in nature. So to do those really detailed chemical analyses of atmospheres has so far been pretty limited to just like the large planets that are really close to their star. And so they're large, which means there's a good chance that you'll be able to see the atmosphere. And close to the star means that there's a good light source. So this is where the James Webb Telescope is really exciting, because it's going to be super sensitive, right? And so hopefully we'll be able to start studying in like some of these smaller objects in finer detail. So access is a huge limitation, and the kind of resolution of the methods. But that's only going to get better and better as time progresses, right? I think when you're thinking about looking for intelligent life, then you're looking for, still it's remote, right? So, you're looking for signals, and the search space is huge. So in my class this past week, we actually talked about the search for intelligent life. And the textbook analogy is that the search space that we've looked at so far is basically like a hot tub in the entire ocean of the world. The search space isn't just looking for like a hey you, it's also looking for the bandwidth, like what bandwidth are they broadcasting on, what wavelength. I mean, there's just like a lot of dimensions that would characterize these signals. And also the fact that civilizations, we don't know how long an average civilization lasts. So our Earth civilization is only 5,000 years old, right? So how long is an average civilization? Does an average civilization last? And our technological age is only like 100 years, right? So our technological age in terms of our ability to send electromagnetic signals off of Earth is super young. So how long will the civilization last? That will also affect our ability to detect it, right? So if it lasted 10,000 years and it's 1,000 light years away and it sent the signal 2,000 or 20,000 years ago, I don't know. It's you have to, the math all has to line up for the signal to arrive at us at just the right time so that we can know it was there. And it's a huge search space.
I've never thought about that. So I think that'll be all for today.
Cool. Well, thank you again for extending the invitation. It's definitely fun to be able to talk about this. It's one of my favorite topics. So I appreciate the chance to talk with the Astronomy Society. And I'm relatively new to NTU, two years in, but those 2 years have both been the longest years and in some way the shortest years, because we haven't really had many opportunities to kind of meet in person and do things. So I hope that with the COVID situation, that your club or your society can get together and do some stuff. And I'd love to join at some point if there's cool observing to do. All right.
So once again, this is the mod that you are teaching right now, right? So it's only available in Sem 1.
That's true.
So I think those of you who have found that this talk has been very interesting can consider taking this module, which personally I most probably will take this the next semester 1.
Awesome. Well, I'll look forward to having you in the class. And anyone who has taken my class will have probably recognized just a couple of the slides. Like I said, I created this class 2 years ago, so it's content that I really love. But this is so you'll see some of the content covered, but in more detail when you take my class. So looking forward to having you, if it works out for you to take. No pressure. Take my class.
As I think the only graduate of that module in the Zoom call now, yeah, everything she said is accurate. Like, sorry, what I meant was her description of the module was accurate in that it goes deeper into many of the concepts that was mentioned in this talk. And in fact, as long as you enjoyed this talk and you, I think an important factor is the level of detail that goes in, because this module is very science heavy. And I personally know that some of those less scientifically inclined people who have taken the module, I wouldn't say suffer, but it was a really steep learning curve for them. So if you love science and how it applies in astronomy, especially in an astrobiological context, you will definitely love this mod, and I really recommend you to take it.
Thanks so much, Raphael.
Yeah.
Yeah, I tried to give everybody a good warning this year that the class was very science intensive. So I hope that served well. But it is an unrestricted elective, and so it's open to anybody to take, but it is very much a science class.
All right. And in fact, I think you did give the warning in my year as well.
Okay, good.
Yeah.
Very good. All right, guys. Well, nice to chat with you again, Raphael, and thank you all for having me here tonight to chat. And yeah, I look forward to engaging with you guys in the future as well.